In looking at our textbook, I'm really trying to make the urinary system palatable, I guess I could say, understandable. In looking at, I'm going to be flipping through and kind of flipping through the text. I can tell you whoever wrote the chapter on the urinary system is a huge specialist on the urinary system and the production of urine. What I'm seeing on page 1222, 1223, 1225 just blew my hair back when I looked at it. We're not going to go into that depth because what I'm afraid of is at an intro college level, you will absolutely fail to see, I think it's called the forest through the trees. It's going to get so convoluted, like the convoluted tubules, that it will make no sense at all. So I'm not going to be dealing with pressures and different concentrations and things like that. I want to show you what happens between the beginning over here at this Bowman's capsule, or the renal capsule, and the collecting duct. And by the time the filtrate, which is what we're going to call it starting here, by this time the filtrate has become full on urine, it's going to get dropped out the papillae into the minor calyx and major calyx, renal pelvis, you get the idea. And then end up in the urinary bladder, which is where we'll finish today up. So how do I get into this? I had kind of given you the run through here. But there's this that I skipped over deliberately, this fishnet of blue. It's a capillary bed. The name of it specifically is the vasa or vasa recta, V-A-S-A-R-E-C-T-A. So as we look at this thing called an nephron, the entire thing is surrounded by, and I'll use the term again, intimately related with the circulatory system. Because what I have to do is I have to keep you keep your feet firmly planted on the ground as far as what way are fluids moving. Are they moving into the blood, out of the blood? Are they going to become urine? Where are they going to go? So, let me find my mouse again. Let me zip from this one, which is the last slide we ended up with on the last video, to this one. Now what this has done is it has kind of cut through. Here's where our little capsule is, or what also is called a corpuscle. This is the very beginning. And then we have our proximal convoluted tubules, and then what we call a loop of Henle, where we got our descending limb, our ascending limb, then we get into our distal convoluted, and then ultimately over here into the collecting duct. I want to take a look at what happens generally in each one of these areas. How do we clean out the blood? How do we regulate what's in the plasma? And how do we make the urine that'll end up wrapping up this lecture, or this video? So, in order to look at this, I've got another slide. We're going to jump to it, and then I'm going to come back to this one again. We've got an afferent arteriole, so our blood supply coming in, and an efferent arteriole, so we've got our blood supply leaving this capsule. Here's what it looks like in the capsule. There's a lot going on. Now here's the capsule itself, and it doesn't really do a lot. It's simply a container. It's like a piece of Tupperware. And we've got our afferent arteriole coming in. There's our efferent leaving. Just ignore this for the time being. We'll get back around to what this thing is all about. But if you look, once it goes in, it looks different. This is still a modified capillary bed, but it looks very, very different. Okay, they don't have the term on here. Each one of these cells, and they are cells, is called a poto for foot site. So it's a foot cell. And what it's going to do is wrap around each one of the capillaries. Now let me see if I can do a drawing like this. It kind of looks like this. I'll put a nucleus in there for good measure. And here's what it's going to do. I don't know if I'm going to be able to do this. Picture my hands as one podocyte, and I've got all these foot-like extensions that are coming off. Now wrap it around in a circle and embrace, completely wrap around one of the capillaries. If you look at how my fingers are intertwined, that's what all of these little things are. These are little openings. They're fenestrations. If you think back, fenestrations, maybe I've heard that. Yeah, when we talked about capillary types. Fenestrations are openings. They're little slots. And what I can do, I don't know if I can do this with my finger, I can loosen up the fenestrations or I can tighten them down. So I can regulate how much of the contents, how much of the chemistry of the plasma dumps out of the circulatory system into this capsule. And then what it's going to do is move on into the proximal convoluted tubule, and that's where all of this is really going to happen. Well, this gets us in the direction of where all of this is going to happen. Let me get the podocyte off of there and work through these different portions of a nephron. Now, one thing that threw me when I first learned about this as a student is we talk about reabsorption. We're going to talk about fluids being reabsorbed. What it means is they're being reabsorbed into the blood, into the vasa recta that you can't see here. The reason we have to do a bunch of reabsorption is in the capsule. I believe it's about 90% of the plasma, almost said plasma again, the plasma is secreted as filtrate. I've seen one number that says we produce filtrate at the rate of about 180 liters per day. Not a big important number. I'm not going to hold you to it, but just understand that 180 liters, I don't have 180 liters of blood in me, of fluid all together. I don't have 180. So what that means is the rest of the trip through here is going to be about reabsorbing, taking materials, water, ions, small molecules back into the circulatory system. So the first important area that we get to is this convoluted tubule. It's all twisted around and has convolutions in it. And it's very, very close to the capsule, so it's proximal. Here's what happens in the proximal convoluted tubules. If you look, they've got a little key down here. I'll just point out what's essentially happening. Water is leaving and ions are leaving. So water and ions, water and sodium and potassium and chloride and important things like that, that without it we don't contract a muscle, we don't have a nerve impulse, we end up dead. So I think it's about 70% of the filtrate that is produced coming out of the capsule is reabsorbed right back into the circulatory system, which is a good thing. Now that leaves about 30% that we have to reabsorb going the rest of the way down through this loop of Henle, H-E-N-L-E. Here's what happens. In the descending limb, we're going to go down and go deep into the kidney. You see there's a thick area and there's a thin area. I really want to focus on water and a little bit on sodium. If I look up at the top, if I look at the thick component of that descending tubule, of the descending limb, it is impermeable to water. Water is not going to go out, water is not going to come in. But once you get to the thin limb of this descending limb, there's a little arrow for water, and I would have drawn water coming out all over the place. So as we go down, as we go deep into the kidney, water is just flying out as we get down here to the bottom of the loop. Now I said I want to focus a little bit, I want to mention sodium. Sodium is going to be one of the big wastes that we find in urine. What happens is sodium is stuck in here, And as we go down and the water goes out, the sodium becomes more and more and more and more concentrated. So by the time we get down to this loop, to where we spin around and we're going to end up going back towards the surface, the filtrate is unbelievably concentrated. We dump all the material out, we absorb water, and we concentrate things like sodium. And urea is another one that we're going to start to concentrate in there. Now we head up. And if you look, there's nothing going on down here. Essentially the water it's impermeable as well. So water is no longer moving out, water is no longer moving in. And by the time we get up to this thick portion of the ascending limb, through active transport, I just got my active and my passive mixed up. Through active transport, where we use energy to actually move things across the membrane, we get a little yellow arrow. This is where we start reabsorbing sodium if we have to, potassium if we have to, chlorine or chloride ions if we have to. And because it's active, this is going to determine how much am I going to allow to be reabsorbed. So if the nephron decides, we're good with the sodium, we don't need to reabsorb it, it's not going to actively pass it across. Just a reminder, this should be completely filled with vasa recta, and it's not. Don't forget that the circulatory system is there through those capillary beds. Now, by the time we get to the distal convoluted tubule, really what we see, I think the most important thing that we see is the secretion of drugs. Up here it's got, what do we have? Acids, drugs, toxins, secretions of ions. So now when we talk about secretion, we're not talking reabsorption, we're talking secretion to where it pulls those things back in. So those have been dumped out of the vasa recta. They're determined here to be waste, and so they're pulled in through the distal convoluted tubule. And then you can see from all of these different nephrons, they're all dumping into this collecting limb or collecting duct. Really what it does is it's going to reabsorb more water. It'll go back and forth with some of the other things like ions, but you're at the very tail end. Really, I think one of the most important things we want in plasma is water. It's a fluid extracellular matrix. So we dump it all out, then we reabsorb water, then we reabsorb water, then we reabsorb water, reabsorb water, reabsorb water. This keeps our blood pressure up. And that's kind of what we want. By the time we dump the filtrate, which is now urine, into those calyces of the kidney, we've taken our trip through. And I would suggest go through the diagrams in our text, get familiar with the terminologies, but don't get buried in the numbers. If this really floats your boat, you go, this is really cool stuff, then by all means, bury yourself in the numbers. But to have a basic understanding of how this system works, I don't think that it is that important for you. Now, I just mentioned blood pressure. And I want to go back to this. This area in here is called the juxtaglomerular, which is Juxta, see if this is too small for you to read, Juxta, Glomer, ULAR, juxtaglomerular apparatus. And it's made up of a couple of different things. The juxtaglomerular cells, which are these in this little tiny wedge in between the afferent and the efferent. And then a series of cells called the macula, M-A-C-U-L-A, densa, D-E-N-S-A. And if you look, well, wait a minute, what's this thing? What's this big tube? This big tube is the distal convoluted tubule. So if I go back a slide, see if I go the right way. You go, wait a minute, that's over here. And here's our little capsule. Why is that all of a sudden over here in the next one? This has been flattened out and ironed very nicely so you could see distal and proximal and whatnot. This isn't just going in one direction to the right, it loops around over here as well. And you throw that in with the macula densa and, or not the macula densa, sorry, with our capillary beds, that really gets confusing. So work with me on this. Here's our distal convoluted tubule, not proximal, with these little macula densa cells and the juxtaglomerular cells that makes this juxtaglomerular apparatus. So let me explain why this is important. One of the last things I mentioned was blood pressure. The juxtaglomerular cells, if they detect low blood pressure, and what a great place to detect it, you're right between two arterioles, they are going to produce renin. I keep wanting to call it renin, but it's actually renin. So renin, this molecule, sorry if I keep stepping out of the view. Renin is going to cause, I'm going to shorten this process up, a molecule called angiotensin to be converted to, I think it's angiotensin 1, and then I think it's the liver converts angiotensin 1 into angiotensin 2, and let me get rid of this. So think back to when we did things like clotting and that cascade. And I said, let's just start out at the very beginning and look at kind of ultimately what we get to. Renin causes angiotensin to ultimately be converted into angiotensin 2. The reason I just erased my arrow is it kind of looked to me like renin becomes angiotensin, and renin doesn't become angiotensin. It just simply causes angiotensin to become 1, and then 1 becomes 2. And you go, well, what's the big deal? Why do we care about angiotensin? Angiotensin 2 has, well, it's got a few different jobs, but really two of the most important are this. It increases the rate of reabsorption of water. Remember, if you're reabsorbing, it's going back into the circulatory system. If I increase the volume, I increase the pressure. That's a good thing. Also, it's going to cause an increase in the production of ADH. ADH stands for anti-diuretic hormone. There's the A, there's the D, there's the H. And what anti-diuretic hormone does is it causes you to be thirsty. So it's going to hold back on urination, and it's going to fire off the signals that will tell you, yep, I'm not urinating or micturating as much, and I am really, really thirsty. One thing I want to mention about ADH, if you drink alcohol, alcohol is an ADH inhibitor. So what that does is it's going to tell you that if I have a couple of beers, all of a sudden it's slowing this down, so I'm not thirsty, but now I have to urinate like crazy. And so if you've ever partaken or you know somebody that did, had a couple of beers, all of a sudden they have to run to the bathroom all the time. It's because they're inhibiting their ADH. Now, what that is going to do is ultimately it will have an impact on blood pressure. And so all of a sudden we've got hormones, we've got, as we'll see in just a minute or so, central nervous system, we've got the circulatory system, urinary system, all working together. And then we throw something in like ethyl alcohol and we start to impact all of these things. So as I've said, we can't just isolate everything and take them all alone. Now, I don't want to go long, and I think I'm pretty much already going long. So there are a couple of things that I want to talk about with the bladder, and I said this is where we would wrap today up. The bladder, if you look at the inside of it, it's not nice and smooth like a bowl. There are little, you can even see the little indents all over here, and it's called rugae, spelled this way, R-U-G-A-E. And if I was to ask everybody right now, okay, why would you think that the inside of the bladder is going to have all of these folds? We would probably come to the conclusion in the next 5 or 6 minutes, or even 2 or 3, that the bladder has to expand. So it has to have give to it, and that's what these rugae are all about. Then around it is a muscle called the detrusor muscle. Detrusor, should be an O. If I had good post-production work, you never would have seen that, but I don't. So the detrusor muscle is what's going to contract to allow you to micturate or to urinate. Also, a couple of sphincters. There's an internal urinary sphincter, and there's an external urinary sphincter. Now location is going to be a little bit different. Internal is going to be closer to the urinary bladder itself. External will be more distal, further away. I think the big difference though that's important to us, the internal is involuntary. The external is voluntary. So if you're sitting in a class and your instructor says you can't use the bathroom, you all of a sudden find yourself really in a bad way. Your internal might have said, it's time to go. So that would release down into the urethra, but now all of your control is your voluntary external. Now, and finally, the stretch receptors on the inside of the bladder get to the point where they tell the pons of the brain. They send the signal back, says to the pons, I'm ready to urinate, here's what happens. I think it's pretty straightforward. The internal relaxes, you will be able to relax the external. The detrusor muscle is going to contract, and all of this waste material, all of this sodium and this potassium and potential drugs and things like that are now going to be released from the body. Your blood continues on just like it was. It's maintained itself, stayed clean, and you've just gotten rid of a lot of waste. That's essentially a look, a very simplified, all of a sudden we have to do everything online, look at the urinary system. So as I said before, please do not get overwhelmed by what we have in this text. Like I said, one of those pages, at least one of those pages blew my hair back, and I don't expect you to delve into and know those numbers. It's not important. So now that I know I've gone long, these two videos that we have just done, I will take my time and I will make sure that if the world of the internet works, it will be 272, not 271 unit. I believe it is now 2 urinary, 1 of 2, and 2 of 2. I hope to have these up. Today is Monday, the 31st, I believe, of March. Last Thursday, I was trying to get one of these up and I fell behind. We have another one coming tomorrow. On Tuesday, well, guess what? These are the 2 urinaries from Thursday of last week, the late 20th of March, and April 1st, I believe it is, that's coming up. So there you go. I hope to have those up. If you have questions, need clarification, please give me an email. Once I get these online and posted, then I've got to start checking emails again. All right, hope you have a great day.